What happens to your brain with 60 seconds of humming? Do this right now. Before a single word of explanation, close your mouth. Find any note. It does not matter which one does, and hold it for 10 seconds. Not in your head. Out loud. Feel where the vibration lands. Not in your throat. Deeper. Behind your cheekbones. Above your eyes. In the hollow center of your skull. The sound is not staying where you made it. It is moving outward through bone at 3,000 meters per second, 9 times faster than it moves through air, and it is entering chambers carved into your skull that you have carried your entire life without knowing what they do. Those chambers are resonating right now. And the gas they are producing inside that resonance is about to change the diameter of the blood vessels in your lungs. You did not do anything unusual. You just closed your mouth and made a sound. The physics did the rest. Now let us understand what that physics actually was. Your skull is not solid bone. It contains 4 pairs of air-filled cavities hollowed directly into the facial skeleton and the cranial vault. Physicians call them the paranasal sinuses. Most people know them only as the things that fill with fluid when they get a cold. That is the wrong mental model entirely. The maxillary sinuses sit behind the cheekbones. They are the largest pair, and they are the ones you felt vibrating most distinctly when you hummed just now. The frontal sinuses are carved into the bone behind your forehead, above the eyebrows. The ethmoid sinuses sit in a cluster between the eyes, small, numerous, arranged like a honeycomb. The sphenoid sinuses are the deepest, buried behind the ethmoids near the center of the skull. When humming produces that resonance you feel inside your head rather than on its surface, the sphenoid sinuses are contributing to it. Each cavity is a separate air-filled chamber lined with ciliated mucous membrane. Each connects to the nasal passage through a narrow opening called the ostium. The ostium is not incidental anatomy. It is the most functionally important feature of the entire system, and the reason will become clear in a moment. The dimensions matter. The maxillary sinus in an adult holds approximately 15 milliliters of air. The frontal sinuses together hold roughly 6 to 7 milliliters. The sphenoid sinuses hold approximately 7 milliliters. These are not large volumes. A tablespoon is 15 milliliters. You are dealing with small, geometrically precise air chambers embedded in bone, each communicating with the outside world through an opening only a few millimeters across. The smallness of the ostium is not an anatomical oversight. It is the feature that makes the system work. A Helmholtz resonator, the class of acoustic resonator these cavities belong to, produces resonance through the interaction between the mass of air in the neck of the resonator and the springiness of the air trapped in the main cavity. The resonant frequency is determined by the volume of the cavity and the geometry of the neck. Smaller neck relative to cavity volume means lower resonant frequency. The ostium geometry and the sinus volume together determine the exact frequency at which that particular cavity will resonate maximally. 4 pairs of sinuses, 4 pairs of resonant frequencies, 4 pairs of chambers tuned by their own anatomy. These cavities are not vestigial. They are not passive. They are not simply drainage passages waiting to become infected. They are resonance chambers, bony enclosures with specific internal volumes, specific geometries, and specific resonant frequencies that are determined by the physics of the air column they contain and the diameter of the ostium that couples them to the airway. You felt the maxillary sinuses resonating behind your cheeks. You felt the frontal sinuses vibrating above your eyes. The sensation of the sound being inside your skull, rather than in your throat, was bone conduction carrying that vibration through the cranial vault at 3,000 to 4,000 meters per second, 9 to 12 times faster than sound through air, and delivering it simultaneously to every bony cavity in your skull. When an oscillating pressure wave matches the natural frequency of an enclosed cavity, the air inside begins oscillating at maximum amplitude. This is acoustic resonance. It is the same physics that makes a wine glass sing when you run a wet finger around its rim. The same physics that makes a pipe organ produce sound from a column of air moving through a tube of a particular length. Your sinuses are Helmholtz resonators, chambers with narrow necks whose resonant frequency is determined by the volume of the cavity and the geometry of the opening connecting it to the outside. When you hum, the vocal cords produce a fundamental frequency somewhere between 100 and 400 Hz, depending on your anatomy and your chosen pitch. That vibration propagates through the laryngeal cartilage, through the hyoid bone, through the mandible, through the temporal bone, and into the entire cranial vault. The skull becomes a vibrating shell. Every cavity inside it resonates. Oscillating air pressure inside the sinuses during humming is measurably higher than during quiet breathing, during speech, or during any other vocal activity a human being produces. The reason is that sustained humming maintains a single continuous frequency input, long enough for the sinus cavities to reach full resonance. Speech disrupts this with every syllable. The frequency changes constantly. Consonants interrupt the airflow entirely. Singing changes pitch with each note. Humming holds one frequency, and the sinuses respond fully to it. Speech changes frequency with every syllable. Singing changes pitch with every note. Humming holds one frequency. The sinuses have time to respond. Not all frequencies produce equal resonance in every sinus. The maxillary sinuses, the largest pair, resonate at approximately 200 to 350 Hz, with exact values depending on individual anatomy. A typical male hum falls between 100 and 200 Hz. A typical female hum falls between 200 and 400. Female fundamental frequencies naturally overlap more closely with maxillary sinus resonance, which is one hypothesis for why some research shows higher nasal nitric oxide output in female subjects under identical humming conditions. For maximum resonance coupling, the physically optimal pitch is a comfortable mid-range note, not the lowest you can produce, which may fall below sinus resonance frequencies, and not the highest, which may exceed them. The note most people choose when they hum without thinking, absent-mindedly while doing something else, tends to fall precisely within the range of maxillary sinus resonance. Whether this is anatomical coincidence, mechanical correlation, or something the body selects unconsciously for maximum coupling efficiency has not been formally studied. The question has not been asked in a laboratory. What is established is the observation itself. Untrained humming without pitch instruction gravitates toward the frequency the physics prefers. You do not need to find the correct note. The note that feels natural, the one that requires no effort, that sustains without strain, is likely already close to the frequency your sinuses are tuned for. Maniscalco and colleagues, publishing in the European Respiratory Journal, demonstrated that humming frequency and amplitude both correlate with nasal nitric oxide output. Louder humming and frequencies closer to sinus resonance produced measurably higher concentrations. The physics favors the relaxed, sustained, mid-range hum, the one produced absent-mindedly in a quiet kitchen, not the one produced with concentrated effort. The absence of effort is not laziness. It is resonance. It is the body finding the frequency that couples most efficiently with the chambers it was born with. What the sinuses produce under resonance is the mechanism that connects a 60-second hum directly to the cardiovascular system. The connection is chemical, and it runs through the mucosal lining of every sinus cavity in your skull. The epithelial cells lining the sinus cavities continuously synthesize nitric oxide through an enzyme called constitutive nitric oxide synthase. This production never stops. During quiet breathing, the nitric oxide diffuses slowly through the ostium into the nasal airway and is inhaled in small quantities with each breath. Production is constant. Delivery is limited by passive diffusion through a narrow anatomical opening. Diffusion is slow. Fick's law governs the rate at which a gas moves across a boundary by passive diffusion, and that rate depends on the concentration gradient and the area of the opening. The ostium is small. The diffusion gradient across it is modest. The result is that a substantial portion of the nitric oxide produced inside the sinus at any given moment is sitting in the cavity rather than reaching the airway. The sinuses are a factory operating at full production rate, with a bottleneck at the shipping dock. There is a secondary implication of this continuous nitric oxide accumulation that concerns infection. Nitric oxide is antimicrobial. It inhibits bacterial and fungal replication directly through mechanisms involving nitrosative damage to microbial DNA and disruption of iron-containing enzymes essential to microbial metabolism. The nitric oxide accumulating inside the sinus cavities during quiet breathing maintains a baseline antimicrobial chemical environment. Humming, by flushing that accumulated gas out and stimulating fresh production to replace it, cycles the antimicrobial environment more actively. The acoustic pressure oscillations also physically ventilate the cavities, drawing fresh air in with each cycle, preventing the stagnant low-oxygen environment in which anaerobic pathogens thrive. Studies of humming in patients with chronic sinusitis have reported improved sinus ventilation and reduced infection frequency. The sinus that is regularly vibrated is the sinus that is regularly flushed, regularly refreshed with antimicrobial gas, and less hospitable to the bacterial colonization that recurrent sinusitis requires. The sinuses are producing a vasodilator continuously. Most of it accumulates inside the cavity, diffusing outward at the rate the ostium geometry permits, because passive diffusion through a narrow opening is slow. Weitzberg and Lundberg at the Karolinska Institute measured nasal nitric oxide output during humming versus quiet breathing and found a 15-fold increase, from approximately 0.2 parts per million during quiet breathing to approximately 3 parts per million during sustained humming. 15-fold. Not a percentage increase. Not a doubling. A multiplication by 15. A magnitude increase that would be considered extraordinary for any pharmacological intervention, let alone a free physical action requiring no equipment and no training. The mechanism is purely acoustic. Each pressure oscillation during humming flushes nitric oxide-enriched air out of the sinus cavity through the ostium and draws fresh air in. The gas that would accumulate slowly during quiet breathing, diffusing outward at the leisurely pace passive diffusion allows, is pumped out actively with each vibration cycle and replaced by fresh air that stimulates further production. The sinus is the production chamber. Humming is the pump. The ostium is the valve. The vibration frequency determines the pumping rate. This is not a pharmacological dose increase. It is a mechanical amplification of delivery, the sinus producing nitric oxide at its normal biological rate. The humming pumping it out 15 times faster than quiet breathing can retrieve it. Think about what that gas does once it enters the nasal airway. You inhale it with the next breath. The nitric oxide-enriched air pumped from the sinuses travels down the trachea and into the lungs. At the alveolar membrane, the thin barrier where gas exchange occurs between air and blood, the nitric oxide crosses into the pulmonary capillary circulation. Once in the blood, it activates soluble guanylate cyclase in vascular smooth muscle cells, increasing intracellular cyclic GMP, which relaxes the smooth muscle and dilates the pulmonary vasculature. This is the identical molecular mechanism of sildenafil, the pharmaceutical vasodilator that generates billions in annual revenue. The molecular pathway is not similar to sildenafil's mechanism. It is identical. One enzyme, 1 second messenger, one smooth muscle response, one vasodilatory outcome, indistinguishable at the molecular level. Your sinuses are manufacturing the molecule. Humming is delivering it to your lungs through acoustic pumping of resonance chambers in your skull. Pulmonary vasodilation has a measurable downstream consequence that follows directly from the physics of gas exchange. Oxygen crosses the alveolar membrane into the blood by diffusion. The rate of that diffusion depends on the surface area of the membrane in contact with blood, and the partial pressure gradient driving the oxygen across. When pulmonary capillaries dilate, more capillary surface area comes into contact with the alveolar membrane. More surface area means more diffusion area. More diffusion area means more oxygen transfer per breath. Each hum cycle that pumps nitric oxide from the sinuses into the lungs produces a brief period of enhanced pulmonary vasodilation and improved oxygen transfer efficiency. The effect is mild. This is not supplemental oxygen. It is not a bronchodilator. It is a modest increase in the efficiency of a gas exchange system that was already functioning. But it is measurable, and it occurs with every hum. If you have ever hummed absent-mindedly, cooking, walking, sitting quietly, and noticed afterward a sense of slightly sharper clarity, slightly more presence, slightly more wakefulness, that sensation is not placebo. It is not the meditative quality of repetitive sound, though that may contribute. It is enhanced oxygen transfer. The pulmonary capillaries were dilated by a gas your own body synthesized and your humming delivered. The pharmaceutical industry has spent billions of dollars developing inhaled nitric oxide delivery systems for pulmonary vasodilation. These systems require compressed gas cylinders, precision flow regulators, specialized masks, continuous concentration monitoring, and hospital settings staffed with trained respiratory therapists. Your skull already contains the production chamber, the delivery architecture, and the pump. The pump is a hum. The delivery system is the sinus anatomy you were given at birth. The molecule is the one the hospital charges thousands of dollars per treatment to administer through industrial equipment. Not at pharmaceutical concentrations. The humming dose is mild, and no one is suggesting it replaces medical nitric oxide therapy for the critical conditions that require precise, continuous, high-dose delivery, neonatal pulmonary hypertension, acute respiratory distress syndrome, and other conditions where dosing precision is not optional. Humming is not that. Humming is the low-dose, self-administered, always available version of a molecular pathway the body already possesses. The mechanism is identical. The dose is different. The accessibility is incomparable. The nitric oxide pathway is the first mechanism. The second is happening simultaneously in a different system entirely, and it begins with a nerve that passes within millimeters of your vocal cords. The vagus nerve, the 10th cranial nerve, descends from the brainstem, passes directly alongside the larynx, and travels down through the chest and into the abdomen, where it governs heart rate, respiratory rate, digestive function, and the broad shift between sympathetic activation and parasympathetic recovery that the autonomic nervous system continuously negotiates. Most nerve stimulation in medicine involves either chemical signals crossing synapses or electrical impulses traveling down axons. What happens to the vagus during humming is neither of those things. It is simpler and more direct. The nerve is physically vibrating. Mechanical deformation of a nerve membrane opens mechanosensitive ion channels, proteins embedded in the membrane that change shape when the membrane bends, allowing charged ions to flow across. Current flows. The nerve fires. The brainstem receives the signal and interprets it as input from the periphery. The heart rate response follows. This is not a subtle or indirect pathway. It is the same class of mechanism by which your fingertips detect texture, by which your inner ear detects sound, by which every mechanoreceptor in the body converts physical deformation into neural signal. The vagus nerve at the larynx is receiving exactly this kind of stimulus during a sustained hum, direct, continuous, rhythmic, mechanical deformation at the frequency of the vocal cords. During humming, the sustained vibration of the vocal cords mechanically stimulates the vagus through the surrounding tissue, not through a neural reflex, not through a hormonal signal, through direct physical vibration of the nerve membrane itself. The vagus is separated from the vibrating vocal cords by millimeters of tissue. The nerve shakes at the frequency of the hum. Mechanosensitive ion channels in the nerve membrane open in response to the physical deformation. The nerve fires. This mechanical stimulation is sustained and uninterrupted for the full duration of the hum. Speech does not maintain it. Consonants stop the airflow. Articulation constantly changes the resonance pattern. Singing interrupts it with breathing pauses, pitch transitions, and note changes. Only humming delivers a continuous, unbroken mechanical stimulus to the vagus for the full duration of a held note. Simultaneously, bone conduction carries the vibration from the vocal cords through the laryngeal cartilage, through the hyoid bone, through the temporal bone, and into the cranial vault. The vagus nerve's intracranial course runs adjacent to the temporal bone. The vibration reaches the nerve from two directions at once, locally at the larynx through surrounding tissue and remotely through the skull via bone conduction. No external sound produces this dual delivery. Only internally generated sustained vibration does. Only humming. If you wore a heart rate monitor during a 60-second hum, you would see the effect begin within 15 to 20 seconds. Heart rate drops, typically 2 to 5 beats per minute, not dramatically. Heart rate variability increases. The autonomic system shifts toward parasympathetic dominance. You would feel it as a subtle settling, a sense of the chest releasing, the shoulders dropping slightly, the jaw releasing tension that had become invisible. The feeling arrives without instruction because the mechanism does not require instruction. The vibration stimulates the nerve responds, the heart slows. The vagal pathway is running simultaneously with a third mechanism, a breathing ratio that humming produces automatically, without counting, without training, and without conscious effort. Humming can only occur during exhalation. The vocal cords require exhaled air flowing past them to produce vibration. Inhaled air flows in the wrong direction. A hum is therefore a form of controlled exhalation, and the inhalation that follows it is a brief, passive recovery. A natural hum cycle produces an exhale-to-inhale ratio of approximately 3:1 or 4:1. The hum extends the exhalation to 8 to 12 seconds. The inhalation that follows takes 2 to 3 seconds. This is not a ratio anyone chose. It is a consequence of vocal cord mechanics. The ratio happens because the physics of the vocal cords makes it happen. This exhale-dominant ratio is precisely the breathing pattern that clinical research identifies as most effective for shifting autonomic balance toward parasympathetic dominance. It is the target ratio of box breathing, of 4-7-8 breathing, of coherence breathing, and of every structured respiratory protocol used in stress management and clinical anxiety treatment. Those protocols exist because the exhale-dominant ratio activates the baroreceptor reflex, the pressure receptors in the aortic arch and carotid sinus that detect the slight increase in blood pressure during extended exhalation and trigger vagal output to slow the heart. Those protocols require training. They require counting. They require weeks of practice before the pattern becomes habitual. They require sustained conscious attention to maintain. People lose count. They get frustrated. They stop. Humming requires a closed mouth and a note. The vocal cords enforce the ratio. The lungs comply. The vagus responds. The person does not count. Consider what this means from a design perspective. If one were to design a breathing intervention from scratch, you would want an extended exhalation relative to inhalation. You would want the ratio to be maintained automatically without conscious effort. You would want it to activate the baroreceptor pathway passively. You would want it to require no equipment, no timer, no instruction, no previous training. And you would want it to be compatible with ordinary daily activity, something a person could do while sitting, while walking, while waiting. The breathing ratio produced by humming satisfies every one of those criteria simultaneously, not because anyone designed it, because the physics of the vocal cords produces it as an unavoidable geometric consequence of how the larynx works. Two vagal pathways are now operating simultaneously: direct mechanical vibration of the nerve at the larynx and baroreceptor-mediated activation from the extended exhalation. Neither pathway alone produces what both together produce. Mechanical stimulation without the breathing ratio generates a more modest autonomic shift. The nerve vibrates, but the baroreceptor pathway does not contribute its signal. Extended exhalation without the mechanical vibration generates a gradual shift that is effective but slow and requires sustained conscious attention to maintain the ratio. Humming activates both through one action. The vibration is automatic. The breathing ratio is automatic. Neither requires conscious maintenance. The person hums. Both pathways fire. The autonomic shift is the sum of two independent signals arriving through two independent neural routes. There is a fourth layer, and it is the most architecturally remarkable of the mechanisms, and the one the research is only beginning to examine. During humming, the partially closed glottis and the sustained exhalation produce a slight elevation in intrathoracic pressure. The pressure inside the chest cavity increases because air is being pushed against a partially closed valve. This pressure increase transmits through the venous system to the intracranial space through a direct hydraulic connection, the internal jugular veins and the dural venous sinuses. The veins connecting the chest to the skull are open tubes. Pressure changes in the chest propagate to pressure changes in the skull within a single cardiac cycle. The result is a gentle, rhythmic oscillation of intracranial pressure synchronized with the humming frequency. Quiet breathing produces this oscillation at lower amplitude. Humming amplifies it through the added resistance of the partially closed glottis. Cerebrospinal fluid, the fluid that surrounds the brain, fills the ventricles, and circulates through the subarachnoid space, responds to these pressure changes. The fluid is not static. It moves with every heartbeat, with every breath, with every change in intrathoracic pressure. Intracranial pressure oscillations synchronized with respiratory mechanics are well documented in neurology. Dreja Kulikovska and colleagues demonstrated that respiratory-driven CSF flow is a quantitatively significant component of cerebrospinal fluid dynamics. Humming amplifies the respiratory pressure component through the added airway resistance of the partially closed glottis, producing larger, more rhythmic CSF oscillations than quiet breathing generates. The glymphatic system, the brain's metabolic waste clearance pathway, depends on CSF movement through the perivascular spaces of the brain. CSF flows along perivascular channels, collecting metabolic byproducts including amyloid beta, and drains through perivenous pathways. Any intervention that increases CSF pulsatility, that moves the fluid more actively through these channels, potentially enhances the clearance of the metabolic waste that accumulates during waking cognitive activity. Humming, by driving rhythmic intracranial pressure oscillations through the mechanics of a partially closed glottis and sustained exhalation, may contribute to this pulsatility. These 5 mechanisms do not all sit on the same evidential shelf, and intellectual honesty requires stating that distinction clearly. The nitric oxide data is strong. Weitzberg and Lundberg measured it directly. The 15-fold increase has been replicated. The molecular mechanism is understood at the level of individual enzymes. The vagal data is established. Mechanical vibration of the nerve is documented. The autonomic shift is measurable in real time on standard monitoring equipment. The breathing ratio is not a hypothesis. It is a physical consequence of vocal cord mechanics that can be derived from first principles and measured in any person who hums. The CSF oscillation mechanism is the most preliminary layer. Intracranial pressure changes from respiratory mechanics are documented. That humming amplifies those changes through added glottal resistance is physically consistent with what we know. That the amplified CSF pulsatility measurably enhances glymphatic clearance during a 60-second hum has not been directly studied. The physics supports the inference. The direct experimental evidence does not yet exist. The viewer should hold that distinction. 3 confirmed mechanisms. 1 preliminary mechanism consistent with established physics. The confirmed mechanisms alone, nitric oxide production, vagal stimulation, exhale-dominant breathing ratio, would justify the 60 seconds. The preliminary mechanism may eventually extend the justification further. The physics supports all 4. The evidence confirms 3. What is striking about this convergence, the reason it cannot simply be noted and moved past, is that every one of these mechanisms activates through the same single action. Not through different interventions stacked on top of each other, through one closed mouth and one sustained note. The acoustic resonance, the bone conduction, the mechanical nerve stimulation, the breathing ratio enforcement, the intrathoracic pressure cycling, these are not separate things requiring separate effort. They are simultaneous consequences of a single physical event, a vibrating column of air enclosed in a skull. Feynman once observed that the most astonishing thing about the world is that it is comprehensible. The humming result carries a version of that astonishment. An organ dismissed for decades as a drainage passage turns out to be manufacturing a cardiovascular molecule that the pharmaceutical industry deploys through hospital equipment costing millions of dollars. And the delivery mechanism is a sound. What you felt when you hummed at the beginning of this discussion, the vibration in the face, the resonance behind the cheekbones and forehead, the sense of something shifting in the center of the skull, was not a curiosity. It was the activation of bony resonance chambers pumping a vasodilator into your airways. It was the mechanical stimulation of a cranial nerve running alongside your vocal cords. It was an exhale-dominant breathing ratio your vocal cords enforced without asking your permission. It was a gentle oscillation of the fluid surrounding your brain, driven by the pressure mechanics of a partially closed glottis. 4 pairs of air-filled chambers carved into the bone of your face. A gas produced continuously inside them that relaxes the blood vessels of the lungs with every hum cycle. A nerve that vibrates when the vocal cords vibrate and cannot stop responding to the mechanical input it receives. A breathing ratio that clinical protocols spend weeks training humans to achieve, produced automatically on the first try by a closed mouth. A fluid surrounding the brain that moves more actively when the skull vibrates gently at a sustained frequency. The mechanisms were running before you knew their names. They will run again the next time you hum, while cooking, while walking, before sleep. The physics does not require your understanding. It only requires a closed mouth and a note. Close your mouth. Pick any note. 60 seconds. The skull already knows how. It is known since before you knew it had chambers. The mechanisms were built in. You were simply never told.